We get going here. All right, so we are live. This is group one. Chris has our first question. He's going to go over it with the screen share. So we'll let Chris take over.
All right. So question one here. So this question concerns epigenetic inheritance. Talks right here about the Engelmann syndrome and genetic imprinting. So we'll get down here to this one. The following cross is done. Female, what are the genotypes and phenotypes of the progeny? So this is what I figured out. I basically worked it with a Punnett square. And I'll show you guys the Punnett square and on my notebook here in a minute. But then, this next question. This question concerns the situation presented in part A. The following cross is done, male, female. What are the genotypes and phenotypes of the progeny? Again, I used a Punnett square. Got this genotype. The phenotypes, all offspring would display the Engelmann syndrome. So yeah, the next part. Suppose a woman has triple X syndrome, which means she has 3 X chromosomes. However, the XX have been deleted from 2 of the X chromosomes. So pretty much it's asking how many bar bodies would there be if you took away 2 of her XX. Because 2 of them are taken away, there will only be 1 bar body. No, I'm sorry. That's supposed to be, there will be 2 bar bodies.
Yeah,
wait.
you're good.
1 bar body, right?
Yeah, I think it's 1. When one's deleted, they only have 1. Yeah.
Yeah. So there's only 1 bar body because the 2 other XX would be deleted, which would cancel out her other 2 X chromosomes, but 1 of them would still be there. For that, I just basically looked through the PowerPoint and reviewed one of the videos talking about this. So it's pretty self-explanatory. I didn't have to work anything out. Suppose you isolate a cell from a female cat who's 2 X chromosomes. However, the T6 genes on each X chromosome are non-functional, while the exist genes on X chromosomes are functional. Basically, it's asking how many bar bodies there would be. I figured out that there would be 2 bar bodies because with there being no T6 RNA to complement the exist RNA, it wouldn't be there to complement it, like I said. So basically, the body would blotch out both of her X chromosomes. So yeah, here's a picture. I guess I can just blow this up instead of turning the camera around and showing you my notebook. Here's the picture of the 2 Punnett squares I used for the A and B part of this problem.
Perfect.
And so that's question number 1. That's how it was done.
Sweet. I'll do screen share, and I'll do question 2, which is right here. Pretty much for question 2 is just to fill in the blanks. So I just went through. If I couldn't do it on my own, the first 2 I didn't remember, so I went back through the lecture notes, reread, and then found the questions that way. And then most of these are found in the notes. I did have a question on the F part. One of the topics discussed in lesson 2 involved the formation of double-stranded RNA molecules. I went back into lesson 2 and I couldn't find it in what I had on double-stranded RNA, but I did see it was mentioned in lesson 3, and I didn't know if she meant it to be what 2 molecules right here, or if that was supposed to be what 2 genes, because in lesson 3 it talks about these 2 genes being part of the double-stranded RNA, but other than that, I felt like this was pretty self-explanatory. That was number 2. And then for number 3, can't remember who had number 3.
I had number 3.
Okay.
So share my screen. All right. Number 3 was again pretty easy, mostly used Punnett squares and stuff. So this is talking about this symptom right here, ornithine. I can't even pronounce that. We'll just call it OTD. And since it's X-linked chromosome, the females are going to have to be recessive, homozygous recessive for it, and the males just need it on their X chromosome to have it. So right here, what is the genotype of the woman's father? What is the phenotype of the woman's mother? So right here we have the father. He just has it on his X chromosome. Since the mother is recessive for it, or homozygous recessive for it, she has it on both of her chromosomes. If the woman has children with another man who does not have OTD, what is the chance that the first child will be a son with OTD? So I worked out on a Punnett square and figured out that they have exactly a 50% chance of having a son with OTD being their firstborn. The genotype of the male with whom a woman could produce a daughter with OTD would be just a male with an X OTD on his chromosome, and then a regular Y one. Consider the cross described in part C of this question. So this cross, or the expected genotypes and phenotypes of the offspring. So the females would have to be this, and the males would be this. Again, I have another picture here I can blow up a little bit to show you. This is how I worked it out. I just wrote XOTDs here and did the crosses, and I just used the same Punnett square for both the answers that needed it. Sorry that my handwriting's a little scribbly and stuff, but that's how I worked it out.
Perfect.
Sweet.
I
got number 4 here then. Get it here.
That looks good right there, man.
Yeah. So for number 4, the presence of beard in the species of goats. Number 4A is right there on the top. So it just shows kind of how I use the big B, little b beard, and then ask for the genotype and phenotypes of the parents right there. And then same thing for B right there. And then we'll go to C and D here. Thing. And then for the on like the sex-linked cells, or chromosomes, not cells, sorry. That's where I changed it to the X and Y.
Mm-hmm.
And then that's what you get there.
Yeah. There you go.
Looks good.
Okay. All right. And then I had number 5 as well. One is right here. Showed for let me read it here. That's different colors in it. I asked for the same exact thing, so I just did the same genotypes, phenotypes, A, and then C here, where it has the very rare symptom for the male. Kind of look this one up. I didn't do it on a Punnett square because it was I can basically take on 2 X chromosomes, and then the Klinefelter syndrome. Do that one. And then here we've got the next ones. Those ones.
Does it look good?
So that's about it.
Perfect. Well, I think that wraps up our group meeting, so should be good.